Sono Baggio, sono direttore generale della stazione sperimentale per le conserve, nonché fondazione. Siamo un'organizzazione nazionale con oltre 3.600 aziende contribuenti. Stiamo lavorando in comune accordo e collaborazione con le associazioni industriali di categoria più importanti a livello nazionale per quanto riguarda sia le conserve dell'ittico, sia le conserve del pomodoro, sia le conserve della vegetali e delle carni. Stiamo lavorando attualmente su 33 progetti, tra progetti istituzionali e progetti europei e regionali, perciò siamo una struttura fondamentale per il comparto conserviero italiano.
Buongiorno, sono Luca Sandei, responsabile dell'area conserve vegetali di SSICA. Parlando di conserve vegetali non possiamo non riferirci ai derivati del pomodoro, ingredienti immancabili delle nostre cucine e nella dieta mediterranea. A questo proposito vorrei ricordare che l'Italia è attualmente il secondo produttore mondiale, con circa 5,16 milioni di tonnellate di pomodoro fresco che viene trasformato in industrie conserviere, con un fatturato totale che si aggira intorno ai 3,5 miliardi di euro, di cui 1,8 che provengono dalle sole esportazioni. SSICA ha da sempre contribuito alla crescita del settore conserviero italiano, diventando centro di eccellenza e sperimentazione all'avanguardia nella ricerca applicata, dedicato alla valorizzazione della qualità e distintività dei prodotti agroalimentari italiani conservati, in particolare dei derivati del pomodoro. L'area conserve vegetali, insieme ad altri dipartimenti SSICA, sviluppa progetti di ricerca applicata per tutti i portatori di interesse della filiera produttiva, delle aziende private, delle associazioni di categoria, delle università e centri di ricerca internazionali, con un approccio dal campo alla tavola. Per esempio, effettuiamo ricerche per la valorizzazione delle produzioni agricole sostenibili, valorizziamo le biodiversità del Made in Italy nei due areali tipici del pomodoro, nord e centro-sud, ottimizziamo e gestiamo i cicli energetici, idrici e il riciclo totale dei sottoprodotti industriali ottenuti. SSICA fornisce supporto tecnologico alle aziende conserviere e a tutto il comparto, pubblico-privato, agendo come Demofarm grazie agli impianti pilota di scala semi-industriale presenti nei laboratori tecnologici di Parma e Angri, vicino Napoli, che consentono di riprodurre integralmente i cicli produttivi industriali. Oltre a importanti innovazioni di processo e di prodotto, ci siamo dedicati alla valorizzazione dei sottoprodotti di scarto industriali del settore vegetale. Per il pomodoro da industria abbiamo messo a punto sistemi di estrazione green per valorizzare le sostanze bioattive.
Buongiorno, sono Chiara Zurlini, ricercatrice del Dipartimento Packaging della SSICA. Nell'ambito del progetto di ricerca europeo Prolific, che vede la partecipazione di un ampio partenariato di centri di ricerca e aziende, sono state sviluppate soluzioni tecnologiche e industriali per il recupero di proteine e altre molecole bioattive di interesse commerciale come fibra, chitosano, polifenoli, dai sottoprodotti di lavorazione agroindustriale di legumi, funghi e caffè, attraverso un approccio di recupero a cascata. Le molecole estratte serviranno alla formulazione di prototipi di prodotto con un TRL finale fra 5 e 8 per il settore alimentare, degli imballaggi bioattivi, della mangimistica animale e della cosmesi. Nello specifico, sfruttando un brevetto di invenzione, SSICA ha ottimizzato su impianto pilota una metodica di processo per l'estrazione di proteine e fibre dai sottoprodotti di lavorazione dei legumi, in particolare ceci e piselli. Parallelamente, nell'ambito del progetto di ricerca europeo Agrimax, finanziato dalla Biobase Industry, SSICA, in collaborazione con numerosi partner internazionali, ha realizzato in Italia una bioraffineria in grado di processare sottoprodotti agricoli e industriali del pomodoro e dei cereali per estrarre composti bioattivi ad alto valore aggiunto, da utilizzare nel settore del packaging e del food per la realizzazione di prototipi con TRL 7-9 in un'ottica di economia circolare. Grazie ai risultati ottenuti dal progetto Agrimax è stata avviata la startup TomaPaint per la produzione industriale di una bioresina naturale estratta dalle bucce del pomodoro da utilizzare come rivestimento per gli imballaggi metallici alimentari. Questo dimostra come sia importante il patrimonio scientifico della stazione sperimentale, che dal 1922, come Demofarm in anticipo rispetto ai tempi, svolge una fondamentale funzione di ricerca applicata e di sviluppo precompetitivo per tutta la filiera agroalimentare.
Okay, so this was the first field visit to the SSICA experimental station in Parma, Italy. We have here with us two of the representatives of SSICA, actually Luca Sandei, who you saw in the video, and Angela Montanari, and now they will share their presentation. Actually, I will do that for them in a second, so that we can continue knowing more about SSICA. So Luca, the floor is yours. Do turn on your camera as well so that people can see you. And
yes, can you hear me? Thank you, Celia.
Yes. Yes, I can hear you.
Okay. First, good afternoon, everybody. Thank you for this opportunity, and it's a great pleasure for me to present you some brief overview regarding SSICA Foundational Research Organization. As you can see, the title of my presentation is SSICA Roots in the Past and Eyes at the Future, and obviously we are talking about research, development, and demonstration for the food processing industry. We talk about safety, quality, sustainability, and innovation. I'm Luca Sandei. I'm the head of the vegetal product area. And obviously, this is just one of the departments here in SSICA. Please, next one. As you can see, the history of SSICA is quite old, but I believe that remain anyway interesting and hopefully up to date. As we see in the video, we are a demo site since 1922. In fact, the REGIA, Stazione Sperimentale per Industria delle Conserve Alimentari, SSICA is the acronymous name of Stazione Sperimentale per Industria delle Conserve Alimentari, was established on July 5, 1922 by a royal decree and began its activity in 1923 in Parma. Obviously, Parma is worldwide known as a province traditionally linked to not only tomato products, also cured meat products, pasta production, and also dairy sector. Any case, for almost 100 years, SSICA has realized research and development projects and pre-competitive studies for the processing Italian industries and all the international stakeholders of processing foods. And last but not least, since 2016, SSICA has become a national foundation for international research with high skilled specific trained researchers and technicians working together in interdisciplinary sectors to obtain the best from fresh sustainability food matters. As you can see just before in the first part of the section of this important connection system, we are talking about the primary sector. Now we are in the middle between the primary and the secondary sector, the industrial sector. We are in this case. Please, then the next one. As you imagine, our main tasks are obviously the pre-competitive industrial application, the analysis, testing, the consistency, technician and technical scientific disclosure, the participation in technical standardization activities. This is very important, but above all, our principal activity of applied research is related towards the more than 3,600 Italian companies that operating the canned meat, fish, fruit, and vegetable sector, frozen products that are distributed in all the national territory. As you can also see, we are in two sites. One is the Parma headquarters, and the second is in the southern part of Italy, that is Angri, in the province of Salerno, close to Naples. Please, the next one. As you can see, now we are talking about the evolution of SSICA, and obviously we have to talk about the past and the present and the future. When we talk about the past, we wish to continue our commitment towards the processing food industries, improving the relationship between European research and development centers, university, and stakeholders' expectation and needs. Obviously, we have studied, applied, in this case, in the last years, bio-based innovation and eco-recycling projects. As you well know, this is a central part of the policy of our European developments. And for some of that, we are patented. For instance, the process, an example will be presented by a colleague of mine, that is Dr. Angela Montanari, that has founded this kind of TomaPaint startup. And she will explain after my presentation. What will be the future? The future will be the application of emerging technologies. For instance, we try to apply extraction of bioactive compounds, bioengineering tools, and other bio-based strategies that are the basis for development of the next sustainability food industry. We are talking about a very sensible industry. We are talking about food. We have to take into account that all the technology that we have to apply will be green technologies. And obviously, we are to exchange information regarding yield, cost, and possibility to apply this kind of application. SSICA, for almost 100 years, has contributed at the development of the Italian agri-food chain sectors, thanks to the continuous activity of this kind of applied research that is obtained thanks to the semi-industrial scale demo plants that are our goal. It's a particular sector of our organization, and this is used to develop the European agri-food research and development sector. Please, the next one. Yes, just to thank you for your attention. Let me particular thanks to all the colleagues of PIC Cluster Conference and for the organization efforts. Please, for any question or clarification, do not hesitate to send me emails or by chat, and for sure I will answer you. Thank you for your attention.
Thank you very much, Luca. And yes, you introduced the next presenter, who is Angela Montanari, who will now show us even more the link between your institution and the market and the company that then will be reaching out to the users. So Angela, would you like to share your own presentation, or do you want me to share mine from my screen?
I try to share.
Sure.
Just a moment. Oh, okay, can you see my presentation?
It's coming up now.
Yes.
Well, it's not showing. I'm not quite sure why, but something happened, but it's not showing.
No, it's not. Could you?
Sure.
Share your...
oh, it's coming up now. Okay,
okay.
Just a bit of delay.
Thank you. Okay. So, thank you. Thank you, Celia, for this opportunity. And my name is Angela Montanari. I am a scientific coordinator in SSICA, and I'm also co-founder and CEO of TomaPaint, an innovative startup founded 2 years ago. The core business of TomaPaint is the industrialization and commercialization of a natural bioresin obtained from industrial tomato processing byproduct. This new performance natural cutine bioresin has numerous high-value applications. The first application that we are focusing on is its use for the production of biolacars for protecting, for coating food metal cans as a natural substitute for conventional petrochemical based lacquer. The starting material is cutin, a component of a cuticle of the tomato peels. The bioresin is extracted from the tomato peels by means of an innovative green and patented process. So we can say that TomaPaint aligns with the principle of the circular economy. Essentially, our business takes agro-food industry waste and turn this into a high value material. Next slide, please.
You're in control, Angela, so you can.
Okay. So, No, perché? Sorry. Non c'è più. Sorry. Okay, and TomaPaint is the end of a pathway starting more than 10 years ago. It is the result of years of challenging research and development. As you can see in this slide, it is a long history that started in the 40s when SSICA patented the original idea. More recently, the bioresin has been studied and optimized in the context of several European projects where SSICA was the coordinator or a partner, for example, from the FP7 Biocopac project to the BBI Agrimax project. Close collaboration with the research system has enabled the development of the innovative technology, first at laboratory level and then at pilot scale. Utilizing Demofarm facility in SSICA. After the startup foundation, we obtained several awards. The most important was last year when we granted the EIC Accelerator Green Deal, a very competitive European call. We obtained a total budget of just under 3 million euros. Thanks to these European funds, we are building the industrial plant that will be operational by next November. The plant will have a capacity of 200 tons per year of cutin produced, expandable to 500 tons per year. We also plan to replicate the plant in the major tomato processing location in Europe by 2025. In fact, we will build a new plant, a second plant in the south of Italy, the important region for the tomatoes, and one more in Spain. In this way, TomaPaint will generate revenues not only from selling the bio-based resin, but also from a licensing package and royalties. Next slide, please. As you can see in this figure, we are located in Parma, in Emilia-Romagna region, where most of the 50% of Italian tomatoes are grown and processed. In the same region, the 70% of the metal packaging manufacturing companies are present. This is a very strategic area for TomaPaint that allows the possibility, the production of a locally sourced lacquer. Next slide, please. Our patented Q10, the bioresin, the first in the market, can be used for a variety of application. As I said, in our entry market application, the Q10 will be the main component of the biolacquer for full metal cans. This market globally amounts to 2 billion euros. Our application roadmap extends to food cans for metal cans for non-food product like general line products. We are evaluating other additional application such as pens for wood substrate, furniture, additive in cosmetics like makeup, or coating for paper substrate, mainly for paper packaging. In fact, consumer concern, market and legislative pressure is driving interest of several industrial sectors in replacing standard conventional oil-based material with a safer and sustainable alternative. This we can consider a total market of about 500 billion euros. Next. The bio-based resin we will produce offer several benefits and advantages. In fact, it increases food safety by removing the risk of contamination of foodstuff with potential dangerous chemical substances such as bisphenol A, BPA, and it meets the demand for sustainable production by using renewable resources and reducing carbon dioxide emission. Finally, our product is exploitable with existing technology, so there aren't barriers to adopt it, and this is a very important point. So in conclusion, after years of research and trials in SSICA, TomaPaint represent a sustainable and effective industrial innovation that could be the catalyst of a new circular value chain. Thank you for your attention.
Thank you very much, Angela. I'm not sure if Enrico Francia wants to say a few words right now, or if we have more exchanges after the second field visit. What would you prefer, Enrico?
For me, it's the same. I would prefer to move to the second part of the field visit, because this part was dedicated to the experimental station in the Emilia-Romagna region. Then the next one will be in Tuscany, and then we will have the complete view from the two sides of the two regions. Maybe it's better.
Yes, perfect. Agreed completely. So Massimiliano, again, if you would like to share the second video so that we go on this visit of the Cesa Demofarm in Tuscany region.
Buongiorno, sono Giovanni Sordi, direttore Ente Terre Regionali Toscane, un ente importante della regione toscana che coordina diverse attività. In questo caso siamo a Cesa, azienda della sperimentazione, in cui stiamo vedendo per la sperimentazione di un nuovo prodotto, il Biochar, un ammendante per il vigneto, che è di alta sostenibilità, è importante per la sua tipologia di economia circolare. In questa azienda abbiamo sperimentazioni di tutti i tipi, sia per quanto riguarda l'agricoltura di precisione, per quanto riguarda la selezione del sementiere, comunque la sperimentazione sui grani e su tutto quello che riguardano i cereali, e naturalmente al servizio delle grandi, piccole e medie aziende toscane di tutta Italia, e con collegamenti internazionali attraverso il progetto Demofarm con Bruxelles.
Marco Locatelli, Innovation Manager di Terre Regionali Toscane, l'agenzia della Regione Toscana che opera nell'innovazione in agricoltura. Il nostro compito è quello di studiare all'interno delle politiche anche europee quali possono essere le opportunità dell'innovazione da trasferire, quindi siamo un centro di trasferimento dell'innovazione verso l'agricoltura toscana. Riteniamo che l'innovazione sia un elemento indispensabile per lo sviluppo delle imprese agricole toscane. Da questo punto di vista, per trasferire l'innovazione abbiamo coniugato questo approccio Demofarm, quindi la nostra è un'azienda cosiddetta dimostrativa che deve in qualche modo riuscire a trasferire questa innovazione agli agricoltori davvero a livello di pari, cioè vedendo in qualche modo anche noi in qualche modo facciamo questo tipo di attività. In inglese si dice peer-to-peer learning. Nell'ambito dell'attività dimostrativa, ad esempio, ci troviamo di fronte a un campo di quinoa, quindi una cultura nuova per il territorio toscano. Facciamo prove anche sull'amaranto, sono cosiddetti pseudocereali e quindi prodotti che possono essere indicati per la dieta, diciamo, gluten free, che in questo momento è molto importante. E in questo centro, in questa azienda, lavoriamo molto anche a livello sperimentale, anche con le multinazionali per la ricerca di sementi, varietà nell'ambito dei cereali e abbiamo prove anche nell'ambito del mais, del girasole. Tutta questa attività ha la sua funzione davvero di essere dimostrativa e quindi di trasferire questa innovazione all'agricoltura. Siamo come una palestra, un luogo di contatto per trasferire questa innovazione.
Sono Francesco Vaccari, ricercatore dell'Istituto per la Bioeconomia del Consiglio Nazionale delle Ricerche e anche vicepresidente dell'Associazione Italiana Biochar. Sono un agronomo, mi occupo da più di 10 anni di capire l'efficacia dell'ammendamento del suolo in agricoltura utilizzando il biochar, uno degli ammendanti definiti carbon negative, sostanzialmente che assorbe anidride carbonica più di quanta ne emetta nel suo ciclo produttivo. La sperimentazione che stiamo conducendo qui a Cesa, in collaborazione con l'Ente Terre Regionali Toscane, è una sperimentazione molto importante per due fattori fondamentali. Il primo fattore è che il biochar è stato dato direttamente all'impianto di questo nuovo vigneto. Nel 2019 all'interno di ogni buco dove è stata messa a dimora ogni singola barbatella sono state inserite 3 kg di biochar per ogni pianta, creando così una serie di filari trattati con il biochar che verranno messi e che sono stati messi in paragone con filari altrettanto non trattati. Questo è molto importante perché è una delle poche sperimentazioni che si svolge fin dall'inizio dell'impianto del vigneto. La seconda motivazione è che ha una durata di lungo periodo perché l'ammendamento con il biochar è una pratica irreversibile, sostanzialmente una volta che si viene dato al terreno non si può più togliere e quindi è importante raccogliere dati sul lungo periodo proprio per cercare di capire l'efficacia e dare delle risposte al mondo operativo. Nei primi 2 anni di formazione del vigneto, perché ancora il vigneto non è ancora entrato nella fase produttiva, abbiamo già avuto un primo riscontro per cui le barbatelle cresciute sui filari biochar hanno prodotto un 20% in più di biomassa rispetto al controllo. Questo fa presupporre che l'apparato radicale si sia approfondito e quindi sostanzialmente che il biochar abbia svolto la sua funzione di ammendante e quindi di migliorare la fertilità del terreno.
Sono Lucia Grossi, tecnico agronomico presso la tenuta sperimentale di Cesa. Oggi ci troviamo all'interno del nostro vigneto 4.0. Si tratta di un vigneto impiantato nel 2019 e costituito da un unico clone di Sangiovese. La peculiarità di questo vigneto è che le barbatelle sono state georeferenziate ed inoltre è stata fatta una digitalizzazione del profilo pedologico del suolo. L'obiettivo del progetto è di realizzare un vigneto seguendo tecnologie di precision farming e applicare quindi interventi colturali sitospecifici, così da avere una gestione sostenibile dell'impianto. Questo progetto è aperto a collaborazioni sia pubbliche che private.
Grazie mille, un video molto interessante. Abbiamo qui con noi due rappresentanti di questo video fatto a Cesa Farm, Marco Locatelli, Francesco Primo Vaccari, che avete visto nel video. Adesso vi do la parola per... Oh, this is Italian. I should be switching back to English. I'm sorry. I'll make it even shorter. I'm going to put your slides up and you're going to be explaining to us a few more things about what you just showed us in the slides. So just a moment, I'll share the screen with you. This is just coming up now. Okay, Marco Locatelli, the floor is yours.
Good afternoon. My name is Marco Locatelli, is the Innovation Manager of Ente Terre Regionali Toscane, a new agency, public agency, and has its headquarters in Florence. The next, please. Terre Regionali Toscane is the agency of Tuscany region, and overall competence is several. For the first, management on the regional agriculture and forestry heritage. In Tuscany, there are 110,000 hectares in forest, where the Tuscany region is the director owner. Is a typical, particular in Tuscany region, is the big forest management of Tuscany region between, in according with the Terre Regionali Toscane. Another competence is agroforestry valorization, and another activity, applied research and experimental in agriculture. And for us, it's very important the approach about the management of the regional germplasm bank and in the land bank for the new farmer approach near the agricultural sector. And another important activity is promoting the demonstration activity in the farm. And the important activity of Terre Regionali Toscane is the demo farm activity. And the director, there are operational branch. We have 3 big farm to demo farm activity in continuous. Cesa Farm is 74 hectares integrated farming, and Alberese 4,200 hectares in organic farming. And the new entry is Suvignano, 638 hectares in organic farming. And the Suvignano come from the mafia organization with the public system to capture the owner of the mafia system. It's a very important place because not only farming activity, but the legal activity for all the citizens of Tuscany citizen. And Cesa is a demo farm since 70, when was created as a regional experimental site. And now Cesa Demofarm is a regional center for testing and transfer to innovation in agriculture. The most important activity in the demo activity. And the main activities: agronomic trials, precision farming technology, cooperation with research institute, university and school, dissemination of research results to the farm, and visit to experimental fields. The next, please. And our approach in innovation and knowledge transfer activity is the system. The system is a multi-actorial approach involvement of local actors and leading companies, and the creation of network between stakeholders, and a new supply chain quinoa made in Tuscany, for example, and bringing farmer closer to innovation. For this activity is very important the network, and our network is composed several parts. The one of the important part is a new constitution, the community of practice in agriculture digitalization. And in this community, there are over 80 members, and the members are private members and the public members. The community of digitalization is work about with us about this approach to develop the digitalization, and the Demofarm approach about this possible to develop this sector. And another part very important in our network is S3 high-tech farming partnership, and other private companies, national and European research institution, area of network, and the Nefertiti network. Is the complex system about the innovation transfer. This is the goal, is innovation transfer. The next, please. And the vision is, for us, is very important to develop about the demo farm is a digital farm where organized demo activity and training activities. It's very important, not only demo activity for the farmer, but training activity for the new farmer is very important about, for us, this approach. And high-tech technology infrastructure, a new demo farm is a place where it is continuous possible to organize tests of high-tech farming, not in laboratories, but in a real farm. We want to work in the farm, in the land, with the new technology. And the farmer-to-farmer learning for us is demonstration farm is a living classroom where agricultural practice can be shared between farming communities. The next. And during the video, you can see about the Biochar trials. And for us, it's very important to long-term environmental benefit and sustainability chain, and the opportunity for a local agri-food chain. But now, Francesco Vaccari speak about these trials. Okay, see you. Bye, thanks.
Perhaps, Marco, I have a question before letting you go. On slide number 4, in the little illustration, I could see an interesting icon. It says texting cow. So could you explain what do you mean by texting cow?
Texting cow in the first?
In the fourth one, number 4.
No, I don't know that.
In the little picture, there is a cow.
Okay, this is the label. This is the label of Terre Regionali Toscane, because, for example, in Alberese farm, there are a livestock, the 400 Maremmana cow, for example, and
okay.
in this sector, but work about the innovation in the whole system to maintain this cow, for example.
Interesting concept of the cow texting.
Actually, it's very different. Probably we check the moment of this cow.
Sure.
Because it's a very big farm, and the animal working is sure. Part of the regional park, Maremmana Regional Park.
It's interesting. It's the future of agriculture, I guess, somehow.
To return, sorry, to the five slide, because the Massimiliano Lelli, possible return to the 5 slide, the number 5.
It was actually me, but so no, I'll just share it again so we see.
No problem.
Here we are on the bottom here, texting cows. I was a little bit surprised.
But the number 5, please. The next.
The number 5. Okay.
5.
Yeah, that's the one.
The next. Okay, record to Massimiliano Lelli during the video. Record. Thank you, sorry.
No worries, thank you.
Okay.
Okay, so I will now share the slides of the next speaker, so Francesco Primo Vaccari. Would you like me to share your slides?
Yes, please, Celia.
I'll do that immediately. Okay, here we are. And the floor is yours, Francesco.
Thank you very much. Okay, good morning, good afternoon to everybody. Thanks to the other very interesting presentation. And thanks to Celia for the perfect organization of this seminar. My presentation will be very short, and I would like to give you some biochar pills. So I don't want to speak about the Cesa field experiment, because in my opinion the video is quite extensive and clear. But in my perspective, I would like to give you some details about the biochar. The biochar, the most common definition, is the product obtained by pyrolysis of biomass when it's incorporated in the soil. Before is vegetable char. When it's incorporated in the soil, became biochar. Next, please. So here with the slide, we have on the left, we have the natural carbon cycle that more or less is considered by CO2 neutral. The plants utilize the CO2, the atmospheric CO2, through the photosynthesis process to organize the inorganic CO2, produce biomass. This biomass more or less is decomposed by the macroflora and macrofauna of the soil, and most of the CO2 that the plants take from the atmosphere return to the atmosphere. So in this case, the carbon cycle is considered to be CO2 neutral, so more or less. On the right side, we have the biochar cycle. If we use the biomass, that the vegetable biomass, to produce the biochar, and the biochar is produced through the pyrolysis, so mass and the other heat, a lot of heat, and the biochar. When the biochar is put on the soil, we sequester CO2, atmospheric CO2. In this case, the biochar cycle is considered as a cycle that is CO2 negative. So in this case, the agriculture make a carbon sequestration. Next, please, Celia. So I am an agronomist. We start with my group to study the effect of the use of the biochar in field, directly in the field, and we started in 2007. We tested the biochar on wheat, rice, processing tomato, grapevine, so in a lot of very different climate and cultivar. What is important for me to say to everybody that the biochar is not a fertilizer, so it's very difficult to give so it's not a fertilizer, so is a soil amendment. So the effect of the biochar on soil and on crops depends by the interaction of climate, soil type, the rate of the biochar distributed in the field. So there is not a unique effect of the biochar. Next, please. So I conclude very briefly with this experiment that we done in 2009 in the vineyard at Antinori Vineyard near Cortona. Is one of the, in my opinion, one of the most important field experiments, because we start to collect data and now we have more or less more than 10 years of field of observation of the biochar effect on vineyard. And now I conclude with this some field. Agriculture is now in the position to provide an effective contribution in mitigating climate change. I'm here, so for any question, I'm here. Thank you very much for all.
Thank you, Francesco. Thank you.
Thank you.
Enrico, the floor is yours.
Thank you, Celia. Just a brief comment. Would like to highlight the importance of the two examples that we have seen, the SSICA station in Parma and the Cesa Farm in Tuscany, about their activity in the research and also in the demonstration of best practices. And this is important also in view of the transfer of the innovations to the agricultural sector, but it can also be viewed as an important action to transfer knowledge, broad knowledge about the sector to the public opinion. And Cesa Farm mentioned this, but I know that also SSICA is doing this. So it is important to spread their activity, their knowledge, also to the public opinion about the agricultural sector, in my view. Now the session will continue, opening a sort of discussion, but also with the help of two other actors in this innovation that are abroad from Italy. One is Professor Peter Sørensen, that is from the Center for Quantitative Genetics and Genomics in Aarhus University in Denmark, and also Clémence Deschers, de Certis Europe from France. And first of all, I want to introduce Professor Sørensen. Peter, are you there?
We can't hear you, Peter.
Maybe the mic is not open.
Sorry. So
okay.
Now?
Yes.
Yes.
I think my mic was muted somewhere else here. Yes. So, good.
Yes, welcome.
Yes, thank you. Yeah, so as you already heard, I come from another part of Europe, from Denmark. And of course, we have other things in agriculture that we are concerned about, but still, I think we also have things that we are common problems. Yeah, and I have prepared a little talk here, or a little presentation. I don't know, Celia, will you share my presentation?
Celia?
Yeah. Yeah. So we just heard that Francesco, he was talking about biochars, and he said that the biochars are not fertilizers. Now I say that in some cases, they actually can also act as fertilizers, because we have done some studies on this, and I agree with him that there are other important properties by biochars that maybe are more important, but I'll show you in a moment that they can work as fertilizers for P and K. P and K are important plant nutrients, as some of you may know, and they are usually applied in fertilizers. So the next, please. So here I show you some results from our studies with different biochars coming from different biomass sources. So normally you use wastes that you treat by pyrolysis, which is a sort of burning without oxygen. And then you get this carbon-rich material we call biochar. And you can see here that they also contain some P and some K, here expressed per ton. If we look at the P, well, we had different biomass sources like wheat straw and chia nut shells, poultry manure and sewage sludge mixed with straw and then treated by pyrolysis. And you can see that the P concentration in these was quite variable, going from 4 kilo P per ton up to 26 kilo P per ton. Normally a crop takes up about 20 kilo P per hectare in Denmark. And that means that if you add less than 1 ton of biochar, you actually apply the normal rate of P for a crop. Also for K, or what we also call potassium, you can see that there's quite a lot of potassium or K in the biochar. So next, please. Another thing that happens when you apply biochar is that you get a change in pH in soil. So in our studies, we found an increased pH between 0.2 points and up to 1.5 units. That means that you get an alkalinization of the soil. It depends very much on what kind of biochar you have, and also on the soil type and application rate. And next, please. I should also say that we have been working mostly with cereals and effects of biochars on cereals here, and we have studied the release of nutrients in the soil as well. Here I show you some results where we measured the availability of the P in these 5 biochars I already showed you. And what you can see for the first 3 ones, the P availability relative to a mineral P fertilizer was about 50% in this study, whereas the last 2 ones, the availability was lower, about 20% compared to a mineral P fertilizer. So it shows that the P is available, and I also showed you before that in some biochars there's quite a lot of P. Next, please. We also studied the availability of the K in these biochars, and here you can see that also that in all cases more than 50% of the K and up to nearly 90% of the K was available in the soil after application of the biochars. So I think even though the main reason for applying biochars is not the nutrients, it's important to have in mind that they contain nutrients that are available. And some of the effects may be due to these nutrients. Next, please. Finally, I will also come with a little warning about that when you make biochars, there is a risk of having some toxic compounds in these biochars. It depends very much on the process that is used. One problematic compound is what we call PAH, which can be very variable and it's toxic. And it's important to take into account this and to use the right technology for making the biochars. If you do this, you can have a low level of PAH and other toxic compounds. Next, please. So now I hope I showed you that, well, by making biochars out of different wastes, this can also be a way to recycle nutrients in agriculture, and especially the nutrients that you have in different wastes. So by this, I say thank you, and questions are welcome.
Thank you, Peter. To be honest, I don't mind the 500 kilometers that are separating us, because 1,500 kilometers is not so much in the view of the climate change context. And I think that the information that we can share, that we can use in the different context, can be applied also to the different context, and we can learn in the different demo farms, in the different university. We can learn about, everybody can learn and can apply for its specific situation the information that we can find with the research and with the applied research. So your presentation was very well focused on explaining certain points that some people is worried about. For example, the contents of pollutants or elements, metals that are inside the biochar. Can you say something about also the raw material that is used to produce the biochar on this point?
Okay, yes, of course. If we talk about, for instance, heavy metals, that's very dependent on what is the input or what is the waste, because the heavy metals, they are there from the beginning, and then they will stay there if you do this treatment. So therefore, well, if you have pure wastes, so to say, with low levels of heavy metals, then you will also have a biochar with low heavy metals in the end. So the quality of the waste is also important.
Yes, of course.
And well, for the other, well, I mentioned these organic toxic compounds. Well, there can be an interaction between the technology and the sort of material you put in. So you should be aware of this. And I'm not engineer, but engineers that construct these this pyrolysis technology, they know more about how to avoid these bad compounds.
Yes, yes. Thank you again, Peter. Thank you again.
You're welcome.
Yes, I was looking at the message. Now we can move to the next presentation, a short presentation that is given by Clémence de Cherf from Service.
Good afternoon. I will...
Bonjour Clémence, bienvenue.
Bonjour. Thank you. Sorry, I won't be able to speak in Italian.
Okay.
Okay, so you have my presentation, perfect. So thank you for inviting me to this conference. I will talk for a few minutes about another use of bio-based innovation and their use for crop protection. Next slide, please. So a few words about my company, Certis Europe. So we are a crop protection company based in Europe. We work on products for several crops, fruits and vegetables and vineyards, arable crops, seed treatments, and potatoes as well. In France, about 30% of our turnover is dedicated to biocontrol products. And it's really a focus for the company because we intend to have a 50% growth for biocontrol for the next 5 years. And we especially have a focus on microorganisms such as Bacillus thuringiensis or Bavaria bassiana, etc., and natural substances. We also have a research facility in France dedicated to biocontrol with screening activities and work on optimizing the factors to improve biocontrol products work. And we develop and register and distribute biological products. So next slide, please. Just a short reminder, when I talk about biocontrol, in France, we have an official definition for that. So it's agents and products using natural mechanism in the context of IPM. There are 4 categories: macroorganisms, microorganisms, semiochemicals, and natural substances. Not all biocontrol products are contain bio-based active substances, but most of them do. In France, the biocontrol market is really enthusiastic. Last year, it represented 12% of the market share, and there is a 30% target in 2030. There is also a very big political push for innovation and development in the biocontrol field. There is a national strategic plan published by the government to really push the deployment of biocontrol products for the next 4 years. It's really a priority sector for public investments and for public calls. And there are also lots of legislative advantages for biocontrol products versus conventional pesticides, such as, and it's just examples, shorter evaluation period to register a biocontrol product, possibility to advertise, etc. So why is there this appetite for bio-based biocontrol products? So it's written really, really short. I can see 3 main reasons. The first one is the food chain specifications. There is a really big increase of food chain specification requiring either less or no residues in final products, and biocontrol products are usually able the farmers to meet these requirements. It can also be used in, for most of them, organic farming. It matches the baby food specification, and these certifications are growing quickly. It also meets the society and farmers' willingness to reduce the conventional pesticide used. In France, for example, it's very pushing in the vineyard sectors. And for companies like Certis, we face greater regulatory requirements to meet in order to register products because of new guidance documents, legislation, and scientific improvements. And also usually biocontrol products have less regulatory restrictions for the use of these products, such as low PHI or no residue situation, etc. We can also explain it by a new approach in favor of integrated pest management. The combination of solutions, not only chemical solutions, but agronomical practices, genetic new varieties, etc., are really growing, and it enables the biocontrol to find its place in this context. And we also have we learn every day, and we have a better understanding of the factors influencing biocontrol products' efficacy, and it enables us to answer farmers' worries or questions about efficacy of this kind of products. Next slide, please. So just a few examples of a bio-based solution developed by my company. For example, we have an insecticide containing maltodextrin, which is a natural substance obtained from cornstarch. So it's used on vegetables to control whiteflies, aphids. And the advantage of that product compared to, for example, a conventional one is that it's a chemical, a physical mode of action. So it's stick the pest and it asphyxiates them. And there is no persistence or residue, therefore it's really compatible with an IPM situation in greenhouses, for example. Another example is a fungicide containing a microorganism, Bacillus aedes. So it's a common soil and rhizosphere bacteria, or also a product under development derivated from sweet lupin seeds. So it's an aqueous extract of germinated sweet lupin seeds. Next slide, please. So to conclude, it's just a really short presentation and a few examples of bio-based products used for crop protection. It's a formidable source of innovation for a sector like ours. We still have work to do improving the efficacy of this kind of product with a better understanding of the mode of actions and factors enhancing the performance of bio-based products, because it's quite different and quite a challenge. There is also work to do because there is a lack of biocontrol solutions available for farmers, especially for arable crops or minor uses. And I'm speaking for Certis as a company. The sourcing of our bio-based products is from I would say primary source, and it's really a field of improvement to go towards industrial by-product source, for example, for our product, to reach even more sustainability and circular economy. So thank you all for your attention, and don't hesitate if you have questions.
Thank you, Clémence. Yes, I've got a question for you, because this was an interesting example in applying the bio-based innovation to the pests and pathogen controls through biocontrols. Based on your experience, what is the influence that the European policies have in determining the adoption of these novel biocontrol products? Or is just a matter to present this innovation to the farmers? I don't think so. That is just a matter of innovation. You also mentioned that the policies, the local, national, and continental policies can have an influence.
Yes, I think it can have a really big influence in lots of sectors. If you think about regulatory sector, we are talking about developing new guidance documents specific to natural substances to enable companies to register products more easily, meeting the safety requirements. For public calls, for example, if you target bio-based products, you can maybe help small companies or startups to develop their products and reach the market. And there is also a lot to do regarding communication to farmers, to technical advisors as well, because we need to communicate more and to explain that sometimes if a product has not worked in the past, it's not because it's not working, but it's because we didn't really know how to use it. We didn't have the knowledge of how to position it, which material use, the temperature or condition, many factors. So we really need to invest in it, and I think European and national policies are here to help.
Yes, of course. The importance of the demo farms again is really crucial in this aspect, to transfer this knowledge and to consciously inform the farmers that there are others technical solutions to control not just chemicals but also biocontrol to avoid pests and pathogen in the field and to improve also the fertilization and the sustainability of the entire system. Yes, of course. Thank you again. Celia, are you there?
Yes, indeed.
Oh, okay. Yeah, just a short comment before to leave maybe the ground to some question or some intervention from the audience. So if you were present in the two parts of this afternoon of this PIC 21 first day, we have tried to give a global vision, in my view, of the bio-based innovation by means of some key examples and how this innovation can contribute to the sustainability, to the whole sustainability of the agricultural systems. And I hope this could have been an opportunity to learn about new realities and also to strengthen the collaboration between the different clusters that are involved in the same topic, that is improve the sustainability of the agricultural systems.
Yes, exactly. Last year, the event was held in France, and there was a very similar approach. So online this last year as well, with visits of specific organizations to understand how they work and to confront the various working practices. The year before, we were in the Netherlands. So of course, we are seeing very different contexts, very different cultures of doing crop production. So I think this really is the point of this event, is to try and show you how we do it in the host country, but also to give the floor to others, like we just heard from Denmark and from France, to have that other kind of angle to things. And I really like the fact that in the presentation from Francesco Primo Vaccari, you said the biochar is not a fertilizer, and then in the presentation of Professor Sorensen from Denmark, you said it is a fertilizer. And of course, the two of you are right. And it's very interesting also to see and to think about these things, to put them in context. And that's really the point of having that sort of event. Francesco, I see you've opened your camera. Perhaps you want to comment on this.
No, yes. No, okay. The biochar, it obviously it can consider as a fertilizer, and the Professor Peter Sørensen has completely right. My affirmation about this is not a fertilizer, because when you use the biochar, you cannot say just before which effect will it have on the crop. I mean, if I use some fertilizer, I know exactly what this fertilizer will do to my crops. In case of the biochar, there is an interaction between crops, climate, type of soil, rate of biochar. So it's not possible just to say the biochar is do these things. If I use the ammonium, the nitrogen or phosphorus or potassium fertilizer, I know exactly what this kind of fertilizer will do. In the biochar, I have to know very well, but I mean, I completely agree with Peter Sørensen.
Sure. No, that was completely clear. It was just semantically very interesting, and I liked it actually a lot. But I think, again, coming back to the main theme of this year's event on demo farming, having a local demo farm where you can test it in the similar environment to where you will actually be using biochar, maybe a few kilometers away, having a local demo farm for a farmer, that might actually be what he needs or she needs to understand the product better. So I'm really hoping that this can be put into context through this network of demo farm that we're trying to build. And our colleague from Holland has shared a link here where she's saying you can be part of this network of demo farm with the project Nefertiti, which was funded to do that, to put them in connection, so that you can use the results of the ones that are most applicable to you. So I think that could be very interesting to help understand what is it going to do to my crop.
No, in my view, the demo farm are the perfect example of the demonstration of what the innovation in agriculture will do in the next year, because I know the agriculture world that is more conservative world, so it's very difficult to introduce an innovation in the agriculture. So in this case, a field experiment can say more than 10 of paper published on Nature or in the very big scientific journal. So in this case, the demo farm, just to introduce at the operative level an innovation in agriculture, is the perfect suitable option to do, in my opinion.
And for the Italian speakers, tomorrow afternoon we have a session that will be in Italian for the Italian stakeholders, let's say, on this topic of demo farming. So I recommend that if you are an Italian-based stakeholder, do participate to this parallel session tomorrow afternoon, where it will be discussed also from an institutional point of view. How do we make sure that this idea is supported also by the institutions and the public authorities? So I recommend that as well. Okay, so I think we're nearly at the given closing time, which was supposed to be 5 o'clock. So I thank you all, speakers and participants, for having hold on, because I do realize it's quite a long session to be online for the whole afternoon. But I hope you found it interesting and rich. We heard, in one afternoon, the whole cycle was kind of covered, from the early stages, talking about the genetics, all the way to reusing some of the byproducts. So, of course, we have only been quite superficial in some elements, perhaps some more specific in others. But do book meetings or email our speakers if you want to know more, if you want to have insights into their results, and if you want to collaborate with any of us. Today, we are, I think, all very open to international collaboration. So we look forward to hearing from you and to see you again tomorrow morning at 10 o'clock. We will start with another conference. Tomorrow, we will concentrate on horticulture and fruticulture, and on the low carbon, low impact innovation for these two types of productions, protected cultures. So hopefully, again, very interesting discussions and talks. We hope that you will be also asking a lot of questions and making comments. I think today's comment section was very busy, so that's fantastic. And do book B2B meetings, and I think I'll leave it at that. And I shall see you tomorrow morning at 10 o'clock. Thank you all again.
Okay, thank you. Bye.
Thank you, Celine.
Bye. Bye. Thank you.
Bye.
Bye.
Thank you, everyone.
Bye.
Bye, everyone.
See you tomorrow.
See you.